11, Chapter 20, Section 1, Introduction to the Protists, which begs the question, what is a protist? Well, folks, we're moving on into a new kingdom, and this kingdom is eukaryotic, much like ourselves. As far as the eukaryotes go, things that have, there we go, we'll just go full size, things that have a nucleus where all the DNA is bound. You can think of a couple of kingdoms that have done that. They've done it fairly efficiently. Fungi, Animalia, and plants, they all do that. But the key point that I want to make very clear to you is that fungi, plants, and animals are hopelessly multicellular. That's what we do. We've specialized to the point where we can't really get away from being multicellular. But there is a kingdom out there, it's very intriguing. It's called Kingdom Protista, and what it does is it can live, these little critters can live their whole lives, if they so choose, as a single solitary eukaryotic cell. Interestingly enough, they can live solitary, if they so choose, but what you'll find in this kingdom is that, well, I like to put it this way, they get lonely and they will colonize, but they do have the ability to be a single cell. Interestingly enough, this kingdom of single cells, if they so choose to be, can look animal-like, and can look plant-like, and can even look fungus-like, and at times it can be really hard for us to tell what we're looking at. We have to look very closely. So we'll get rolling with the Kingdom Protista. So I've kind of given this way a little bit. What are protists? Well, they're eukaryotes, and that term is an interesting one. And I keep pointing out to students, you gotta really know what these terms mean. Eukaryote comes from that word we've talked about a few times this year. There we go. Meaning true. And karyo, more or less meaning nucleus. We didn't see that in our friends, the bacteria. In fact, they were so small that they were on par with size in being similar in size, I guess, to a nucleus. They're really, really tiny. Remember, a lot of our cells, I always point out, a red blood cell, for example, a typical one is about 30 micrometers, 30, 40 micrometers. It's not very big. Well, imagine how small the nucleus is. Get down to a really tiny size and you're approaching the size of bacteria. So, getting back to the protists, they're not plants, and they're not animal, and they're not fungi, but boy, sometimes do they try to resemble them. There are quite a few of the protists, 200,000 species in all. Interestingly enough, I mentioned them being strongly capable of being unicellular, but they can buddy up if they so choose. And at that point, we start looking at sort of the beginnings of what it means to be multicellular. Not to worry, we'll make this all make sense. We'll start out with some really simple examples. There's a lot of protists that you know, probably right off the top of your head. You know about amoeba, and you know about parameciums, or paramecia, most correctly stated. Let's stop for a second. We'll take a look at those now, and then we'll come back and get a little bit more into examples of Kingdom Protista. So I'll just pop out a reflection for just one sec. And go back to computer mode here. Okay.
Protists are abundant in water and on land. Most, like this amoeba, are single-celled. Its false feet or pseudopodia are used for feeding and locomotion. Volvox is a colony of single-celled organisms. Special reproductive cells give rise to new individuals. Paramecium is covered in tiny hairs called cilia that beat in time to produce movement.
Now, Paramecia, that's probably one of the most common ones. You really want to be aware of these little cilia on the edge, and these are quite quite advanced. They're the same cilia that we see in our lungs, and you can think of them as almost tiny little hairs that beat on the edge of the cell, and they give these cells locomotion. In our cells, they help to actually clear out things, mucus, etc., to keep our lung cells healthy. If you're a smoker, you can really do damage to the cilia, natural cilia in the cells of your lungs.
It uses a structure called a vacuole to engulf and digest food. Paraneema, however, can manufacture its own food and uses its whip-like flagellum to propel it forward.
So that was kind of neat. And this sort of pause point here in the slide, you could see all kinds of things. We can see Paramecium, examples of Paramecia here. This one's red because it's been eating little red pigmented yeast cells. And up here, we see some beautiful colonial single cells that have gotten together and formed these balls. We'll see them in class. They're known as Volvox, very attractive. When we look at Paramecia, we can see plant-like ones and animal-like ones. And down here in the corner, yes, right down there, looks pretty slimy, you'll even see slimy ones. So it's kind of neat because this shows them sort of all at the same time, right? Where you've got animal-like ones and plant-like ones and even fungus-like ones. But because these critters in Kingdom Protista aren't specialized multicellular critters like us, they don't belong in those major kingdoms. They're not, by nature, a multicellular group. They can colonize together, but they're not like us, where we've got all these incredible specialized cells. They were just getting started. They're not as advanced as we are. Okay, so I'm going to pop out. I got a little bit more for you here. And this is a neat one. It talks about what are the protozoa. I'm going to come back here once we get a chance to look a little further. I'll make this animation available on the website. It is really, really good. But for now, let's get a little bit more note material out of the way. All right. When we think of the first eukaryotic organisms on the planet, protists were the inheritors. If you think of where bacteria would have been going, there must have been some types of single cells that decided to bind their nucleus, take their DNA, and instead of having that strange nucleoid region that we saw in bacteria, the bacteria were just kind of sloppily putting their DNA threads kind of in a central region. But at some point, let me just undo that for a sec, the threads of genetic material were encapsulated inside of a nucleus. So I'll draw this nucleus forming around the genetic material. And it would be an advantage to do this because if your genetic material was bound in a nucleus, when a cell went to divide, it would be very well packaged, wouldn't it? It makes sense to set up a nucleus. So I'll just put a little note here, a nucleus. I have to do on there we go. Just sort of do it on two levels. My apologies there. This is where cells eventually went. It's not the Monarans, or more correctly termed bacteria, aren't on the planet anymore. If you think of the evolution of critters on our planet, there is mutation and there is change. And at some point, the protists came along and they were larger, more advanced cells that had decided to package up their DNA, and they're just the next stage in the evolution. You could think of them as moving the whole picture forward. So to say that they descended from prokaryotic cells really makes a lot of sense, because the first cells that we find on the planet are indeed the prokaryotic cells. Interestingly enough, proof that the prokaryotic cells were around. If you think back to some of our earlier lectures, we were talking about stromatolites and cyanobacteria, and we found the archaebacteria. We know exactly how old these are, and there's plenty of evidence as to what they've done to sort of move the planet along. We even talked about the fact that some of these original microorganisms became the first chloroplasts inside of plant cells and became even our mitochondria, our energy-producing mitochondria that are inside of our cells now. And if you remember back to the evolution unit, we called that the Endosymbiotic Theory. All right, so protists, if we've got 200,000 plus critters to sort out, let's put them into groups. Remember, biologists are always keen on setting up the grand library of life. Where does everything belong? Well, we've seen heterotrophs. That term has come up quite a bit. So if something has to eat something else, we say it's heterotrophic, and that's a definite animal-like characteristic. Okay, so one that you know and love, Paramecium, right, would fit into that group. Now, not to be outdone, and I'll even use a green color for you. Let's adjust that so it's a little bit more transparent. There we go. We have some of these critters can actually photosynthesize, so we say that they are plant-like protists. And you're going to meet a neat little one called Euglena. It's very small, and I'll tell you right now, when we do the lab, you're barely going to see them on medium power. You'll have to go to high power and watch them dart around. So they're the best examples of our photosynthesizers, and they work really well in the lab. As we move on, we have fungal-like ones. So let's go with a brown color and take that down a little bit. There we go. So we've got, ooh, that's not what I wanted. And we've got our fungal-like critters, and these ones are our very important decomposer group. So I'll highlight that in brown, just like kind of like mushroom brown. There you go. They're decomposers, and these ones tend to be there's some parasites in that group too. So I'm gonna basically what you should label on here is that you've got animal like protists, and you've got plant like, and you've got fungus like. And that's how we'll go about classifying these and getting you to know them. There we go. Give you a chance to scribble that down. If you want to look at it this way, we're classifying them based on their eating pattern. We can also classify them not just based on the way that they eat, but we'll look at the way that they move. Very helpful. All right, that takes us to the end of section 20-1, but what I'm going to do, this topic just leads into the next one. I'm going to pop out and go from here straight into 20-2, because we've said, well, hey, there's animal-like, plant-like, and fungus-like protists, so let's begin with the animal-like group. So again, we sort of have our introductory slide. I'll give you a second to pull out your notes pack for 20-2. And we'll go on to slide number 2. So there's a little reminder here, since it's sort of it's reviewing an element of the previous lesson. These are eukaryotes, but they're not kingdom Plantae. They're not kingdom Animalia, and they are not kingdom Fungi. But boy, do they have a lot of characteristics of them. This is a really important point, and I think it keeps getting messed up every time people talk about the protists. Most protists, here's a little most, are unicellular. Okay? A lot of these protists, like Paramecia, sure, they'll whiz around and they'll feed on clumps of vegetation, and they'll eat anything that they can basically catch. A vast majority of the protists are unicellular, but as we've been investigating the protists, we found that there's arrangements where they can get together and they look multicellular. Well, that makes a lot of sense because the first advanced cells would have been sort of solitary, a lot like the bacteria would have been. But then coming together and working as a group, they learned that there's some advantages in coming together as a collective, right? And my grandfather used to say it best: many hands make like work, or many hands make light work. Ooh, there's a tongue twister. It's just an advantage to come together as a group. And you see this even amongst multicellular organisms today. A school of fish, a pack of wolves, right? A pride of lions. So that trick has never stopped. But Paramecia do not have to be multicellular. In fact, they do very well just hanging out by themselves. Okay, so this slide is redundant with what you saw just a moment ago, but let's call them exactly as they should be classified. You've got your heterotrophs, so animal-like. You've got your photosynthesizing plant-like ones, and your decomposers, which are fungus-like. So let me highlight that, like that. Give you a good idea of the three sort of major groups that you're looking at. And a word to the wise, you're not going to see a bacteria-like group, okay? At this point, if we go back in evolutionary history, the protists look like they're sort of a launching pad, don't you think? Where there's a single cell and this one's acting heterotrophically and this one's going off to be plant-like and this one's going off to be fungus-like. Interestingly enough, right, now there are multicellular animals on our planet, and multicellular plants and multicellular fungi. But the unicellular protists are still around. Well, and some of the ones that get together in colonies and act in a multicellular way, they're still around because they've been very successful at what they do. Okay, so at this point, I'm going to buzz out. I'll take a little introduction to our friends here. We could start with one of the most common ones that we look at, which is an amoeba. So on your sheet, write down that this fellow here is an amoeba. Now there's a couple spellings. I'm preferential to sort of a conservative spelling. This one here with the OE. And your amoeba, this, you might as well just say that this is on the test, okay? A couple of things you have to know about amoeba is that they move in a lot of respects very similar to our white blood cells. They kind of ooze their way around. So let's go meet them. We'll come back to the diagram. And let's see what we see. Okay, now this is a neat little diagram here. It's a little flash diagram, and this up here is a ciliated paramecium, and you can see the cilia really well. But this amoeba right here, we'll meet that, has some pretty neat structures. First of all, it has something down here that we call a pseudopod. It looks like it's reaching out with its foot. Like you can see here that it moves by streaming in one direction. Okay, so that's pretty important. So let's see. I'll go back through history, and here's an amoeba in motion. Very, very interesting. And I showed this on Friday, but it's worth seeing it again. And let's see. Let's pause that, and we'll get. Ooh, I got kicked out. That's all right. It's my computer. I can come back in anytime I want. We'll go to full screen, and we'll watch this. So you can see the cytoplasmic streaming. I'll just pause. So this is the cell membrane, and as it streams, it pushes its cytoplasm into these little extensions that we call pseudopods. And there's some nice crystals in here as well. These are very pretty. And these are hunters, right? These are heterotrophs. Amoebas will find things and engulf them and eat them, which is, in a lot of respects, what our white blood cells do. Our white blood cells are very amoeboid. You can kind of see where they got some of their heritage. So you have to know that these are called pseudopods, because if you don't, it'll drive me crazy. Pseudopod is a very neat term. It means a false foot. Zoom in on this again. There we go. So this chapter is all about diagrams, folks. Get used to it, get to know it. When amoebas extend, we have the pseudopods, and when they take something in, they form a little baggie. And they basically swell around their food and they take it in through a little process that we know, it's kind of a fancy word, it's called endocytosis. And when the food comes in, right, it passes through the membrane, it becomes a little baggie like this. And they're called vacuoles. Now, depending on what's in the little baggie, you just kind of give it a name. If the amoeba ate something, you call it a food vacuole. If the amoeba had a drink of water, so to speak, inside the baggie would be water, and we call it a water vacuole. And amoebas can push water out of their membranes and literally go pee by pushing what's called a contractile vacuole up against the edge of their membrane, and they release water. And you have to do that because if you have too much water, if you take on too much water, you can burst. So think about it. As humans, we get rid of water. Our kidneys are very instrumental in that. And very important to point out over here is the nucleus. So the nucleus, there you go, organized region for DNA. Cytoplasm, the gel that everything is resting in. And one at a time, this is neat. We can label each one. You can see those pseudopods moving around. Here's that contractile vacuole, and it releases water. They're very handy. Cell membrane, there we go. Quite tough as it turns out. Food vacuole, that was a neat little animation. You can see the food that was brought in. The nucleus is over here. Cytoplasm, right there. So that's a good little overview, and it's nice that I'm capturing this on the video because we'll be able to look at it as many times as we want, I suppose. Now our friend the Paramecium, take a look at him. Paramecia are interesting creatures. Well, I wanted you to get an overview of it at this little critter because this is a little bit different. You can actually see it almost looks like he's smiling in there, right? There's his mouth, and no, these aren't eyes. The Paramecium has a large and a small nucleus. The large nucleus is the normal one, and the micronucleus is kind of like a backup. It's pretty smart. It's like it's making a backup file of its genetic code. And it's got an anal pore where it can poop, release its waste. Here's the contractile vacuoles in the way that I'm a little happier showing them to you. They look like sort of like these little stars, right? Because when they squeeze, they can squeeze the water out. So a little bit better of a representation. Food vacuoles, right? These little dark spots in here. Cytoplasm, everything's floating in it. And the cilia, which are beating and giving it mobility. So we'll come back to this because there's a couple of neat little things here to talk about. In particular, this one called trichosis. They're like little spears, but I don't want to get too far ahead. And you know me, I probably will. Okay, so back to the boring flat diagram. Now that you've seen these structures, you'll have an opportunity really to get to know these diagrams. You can see that this always drives students crazy because sometimes the structures are drawn a little differently, right? The food vacuoles here are green, and the contractile vacuole isn't exactly drawn to specs. The way I would draw the contractile vacuole is I draw it like a little star. You makes it a little bit more obvious. And that's something you'll see come up in many of our diagrams. In fact, we have a package for you for this unit to, well, yes, get you guys coloring and getting you to know the diagrams, because we have to work you over. So there's 4 major phyla. So if you think about it, if it's Kingdom Protista, and if you recall, you've got Kingdom, Phylum, Class, Order, Family, Genus, Species. Holy, that's a mouthful. Kingdom, Phylum, Class, Order, Family, Genus, Species. We have to take the Kingdom Protista and break it down into its major groups. So for the animal-like ones, there's some major phyla. For the plant-like ones, there's major phyla. And you guessed it, for the fungus-like ones, there are major phylums. So 4 phyla. So if you're studying, you're saying, oh boy, I gotta know, there's 4 phyla I'm responsible for. And you should be able to write these down on a pad of paper and just know them. First phylum you need to know about, zooflagellates. Here's a hint, they've got flagella. So zoo just means animal-like, which I think is pretty redundant since we're already looking at animal-like protists. But we'll say that these guys have flagella. Right? We come down to the ciliates down here. Cilia are just short flagella. So this group, Paramecium, would fit into this group. So we'll say that they're ciliated. Right? Our sarcodines have amoeboid motion. So we'll just say amoeboid for short. And the sporozoans, well, I always underline spore here because I think about spores that float through the air. For example, I think about fungal spores. They really can't move on their own. Something else has to move them. So these sporozoans are non-motile. They're very interesting because, well, one of the worst ones has a lot to do with malaria. Let's put it that way. We'll talk about it. And really, unless this critter gets in your bloodstream, it can't really move. It has to be moved by other things, vectors that kind of do the job for it. So, animal-like protists, classified by how they move. And that's an important point to make. It's a pretty simple way of classifying the animal-like ones. So I'll give you a chance to copy that down. We start by focusing on the animal-like ones, and then later we move on into the plant-like ones and the fungus-like ones. The fungus-like ones are fascinating. You could spend days just talking about the fungus-like ones. But we won't, because we don't have that much time in the course. So the zooflagellates, let's zoom in. When you look at the zooflagellates, let's just use a nice red color. You've got these, this is what distinguishes flagella, right? Long. Cilia don't do this. There we go. And sometimes there's combinations. You'll see that they can have cilia and flagella, but if they have flagella, we say that they're belonging in the zooflagella group. Okay? Animal-like ones with flagella. Oop, I hope I didn't go too quick there. I'll give you a chance to write that down. They swim using flagella, cruise around. Now flagella, it's interesting because they almost whip. It's very different than bacterial flagella. My description of bacterial flagella is it runs like a prop on a boat motor. Now these are different. These are very much like, well, how sperm would move, which is a back and forth motion like this. Now cilia and flagella can both be used this way, this back and forth. This back and forth type motion. You get the idea. Almost like the way when you watch a salmon swim back side. Cilia and Flagella do the same thing. It's just that flagella are longer. Sarcodines are basically amoeba, and when we describe them, we say that they have cytoplasmic streaming or amoeboid movement. These pseudopods, and since I have my red color, a pseudopod can form, there we go, from any extension of the cell membrane. It just depends if our little critter here feels like flowing in one direction or another. So it's temporary cytoplasmic streaming because these fake little feet that they're producing, and this particular example isn't showing a lot, but I'll just draw it to the side. This would be a pseudopod. That would be a pseudopod. You get the idea. It's not like it doesn't flow back together. It's just creeping around with an oozing-like motion. And it's sniffing around and hunting down something to eat. So these pseudopods are really how it puts its body out there and just sort of pulls itself along, right? Interesting sort of crawling-like motion. Pseudopods. So if at any time you need to pause or rewind me, you can always feel free to do so. We're going to move along. As soon as I zoom in on that food vacuole, that's pretty cool. You can actually see the contents inside of it. And the nucleus is pointed out. It's a very granular material. This is an exceptionally, this is great microscope work where you can see up close the detail in this sarcodine protist. And if I say sarcodine protist, you just say amoeba, because that's a good stock example for you to have in mind. So there we go. They're flexible, they're active cells, and those pseudopods are sort of thick and they extend out. So we refer to that as cytoplasmic streaming. Amoeboid movement, again, your cells do this, specifically your white blood cells, which gets you thinking kind of about the evolution of those cells, right? Where did our white blood cells learn the trick to be amoeboid-like? This is all fascinating. When you move on, if you decide to carry forward in biology, ask those questions. When you get into histology classes, when you're studying tissue and things like that. These are great questions to ask of your professors. Okay, so I'll give you a moment longer to scribble this down. If you need to, if you're at home watching this, you can always press pause. If this is being presented in class, well, you can always press pause or just go back a little bit, whichever you need to do. So redundant slide, we're back to the structure of the amoeba with its pseudopods and food vacuole, nucleus, and contractile vacuole. Contractile is okay. That's the kind of vacuole which will squeeze with the cell membrane and let water go. But these things can also have water vacuoles. They can also take water in. So depending on what's inside that little baggie, that little inside the cell, is generally how you name it. If it's a vacuole, it could be one of many varieties. Okay, so our ciliates. Now, when our lab samples come in, you're going to see all kinds. This one in particular, right, is one we call Vorticella. It's got all these great names, I know. I need to pick a different color so you can see this. They have rings of cilia, and interestingly enough, they're beating and they're creating a current to try to bring food in so that they can basically eat. So they'll grab Paramecia or any critter nearby that's unfortunate enough to get pulled in, right? And they have stalks, so these ciliates can actually be fixed in place. But in a way, if you just look at it, you say, oh, that's just filter feeding. I've seen that before. And in fact, filter feeding is a trick which uses cilia throughout the animal kingdom. cilia are very useful for creating these currents and for grabbing microorganisms for the purpose of feeding. And that's an advantage with cilia because flagella are quite large, right? A little bit more of sort of grand movements, but the cilia, they're so small that they turn out to be really, really useful. Not just propelling the cell, but helping feed it too. So the stalk cilia that you would want to know would be our friend the Paramecium. So let's highlight them. There we go. And they're pretty big. When we go to take a look at these under the microscope, under medium power, which is about 100 times magnification, you'll be able to see them quite well. We'll add something to the water, their watery environment rather, to help you to see them a little bit more, to slow them down because they move really quickly. But their cilia are really kind of, I'm exaggerating it here, but it's around the outside like this. You get the idea. But if you look close and you look kind of under the skin, under the membrane, you wonder, well, what is, let's switch up our colors here. You wonder, well, what's this? What is that stuff just underneath? And it kind of carries on around the cell. What is that? Well, it turns out these little guys are hunters, and what they're doing is they've got these little sort of harpoons, and they're just under the skin, and they're called trichocysts. I know, fancy word, right? Trichocysts. I tend to remember it by thinking of the first three letters are tri, like trident, like little spears. And sure, these Paramecia will move around with their cilia, but they can also fire little harpoons. And these little harpoons, well, basically are damaging to things that they want to kill and consume. And here it is. So if we zoom in here, all right, now I'll zoom back out in a moment, I promise. There, you can see they're just lying under the skin, and it always reminds me of how things work with jellyfish too. Jellyfish have a similar sort of tactic, where if you've ever encountered a jellyfish, literally contact with it can release its trichocysts, where you get jabbed with these little barbs, and they can release chemicals into their prey. Well, the idea of these little tiny harpoons, well, it showed up in Paramecia. It's been there for a very, very long time. Now, this diagram is one you would really want to know. So let's put test on this. And you will be filling out and coloring many diagrams to make sure that you know the structure. The oral groove is essentially the mouth, leading down to the gullet so the food comes in. Right? So I'll draw this like this. And at some point, waste products have to leave, so you've got an anal pore. Let's switch colors. Its nucleus is here, but in Paramecium, in Paramecia, you'll see the micronucleus, or the backup for the genetic material. So the DNA, if it helps you, is in those structures there. Contractile vacuole looking like a sun, you know, there's one there, and there's also another contractile vacuole, CV, right there. The food vacuoles here are being represented in green. I don't know how much that helps you when you're dealing with a black and white slideshow, but it's not like we're not trying here. And there's these little tiny things, they call them lysosomes. Put a little note that those contain enzymes. I'll talk more at length about that later in the course. But if you bring in food and you've got lysosomes, the lysosomes, I have to set my computer not to go to sleep so fast. The The lysosomes give you some enzymatic digestion, right? So lysosomes are, in a way, digestive little helpers. And it's pointing to these little tiny little dots here. And there's lots of them. They're also known as suicide bags. The enzymes in them are so strong that if they got out, they would start digesting the cell. And in fact, when we hang meat, let's say that you've shot an elk or a moose or whatever, those lysosomes will pop, and the meat literally cures in a way while it's just more or less hanging in the shop. And that's natural. That's what's supposed to happen as cells decompose and die. We also have our sporozoans. Now, our sporozoans, as mentioned before, they don't move at all, right? They're parasitic. And this is where I'm going to talk a lot about critters like, well, the disease is malaria, but it's caused by these horrendously parasitic creatures. Sporozoans, think of the word spore. It's like, can only really be moved around by something else. These sporozoans are parasites, and we find them inhabiting the digestive tracts of worms and fish, and it gets really nasty when some of these sporozoans jump from host to host. In some cases, with the itch, for example, and you might have heard or much less experienced the itch, the sporozoan, when it gets into the water, can use one host for a little while until it jumps to another host, right? So we'll go into that, but sporozoans themselves, they're just looking to get inside of a body and to cause some parasitic damage. They can't move on their own, so there's no cilia, there's no flagella, nothing. They really just have to get, preferably, into a bloodstream and get carried along. Now, this one here is, depending on the mosquito that you're looking at here, there's two stock examples that we discuss in this course. We talk about an African sleeping sickness, and we also talk about malaria. So when you look at malaria, it's caused by a person being bitten by the Anopheles mosquito. So the Anopheles mosquito, this happens in moist climates, in warm climates. If you go to Africa, you have to watch out for this, right? But there's many moist, warm, equatorial regions on the planet where you really have to watch out for bug bites. It's bad enough that the mosquito's sucking on you, but what if there was something inside the mosquito that was worse? Turns out that if we go inside of the mosquito's gut, that it's infected, that it's got a parasite. And there's a little cyst right here. A lot of these parasites will form cysts in the tissue, and they'll bust out through an infective cycle. It's sort of a never-ending repeating cycle. What happens with the mosquito is these cysts contain these little parasites, and these parasites, known as sporozoites, get into the mosquito's salivary glands, and guess what happens? The mosquito bites you, and the infected saliva from the mosquito enters your body. Mosquitoes inject a little bit of their saliva, of course, because they're sucking on you, and it also contains a chemical that keeps you bleeding freely. Mosquito bites are itchy for this very reason. There's this annoying chemical. Now imagine if there's not just the chemical, but there's what we call a sporozoite parasite. So these little parasites that really only move around in fluids. Those, look at this a little more closely. If those parasites get into your bloodstream, then they've got a free ride just about anywhere they want to go. And since our blood is universally filtered by our liver, let's go with a purple color. If they can get through our bloodstream, then they're going to end up at our liver, and that is their preferred home. Now, this disease, known as malaria, caused by Plasmodium vivax, the critter has a genus named Plasmodium, species named Vivax. It goes through different stages where it can look a little bit different. Here we refer to it as it's broken out from the mosquito in its sporozoite form. Once it hits our liver, it does a little maturing, undergoes some changes, and that mosquito can gestate in our liver and hide out. And this disease is one that returning soldiers will get, and it can be a 40-year disease. You can experience outbreaks of malaria. The disease is very horrific because you can go through periods of extended fever and chills and fever and chills and fever and chills as you go through an infective cycle. We'll go over that more, but this slide is just getting you to understand that this is a complex life cycle because this thing has more than one host, right? You've got your mosquito here, so this is the first host, and we become a host, and the mosquito gets reinfected by consuming infected blood. So it's sort of a never-ending cycle. The mosquito gives you the disease, and the mosquito gets infected by consuming the blood of somebody who's got the disease. So malaria is one of the world's most serious infective diseases. As they say, it affects 2 million people per year. The best way to deal with malaria is to get the Anopheles mosquito under control, because that's the vector, that's the carrier of the disease. A lot of things have been used to try to wipe out the Anopheles mosquito. They used a nasty little powdered chemical for a long period of time known as DDT, and no, that's more than a wrestling move. DDT was literally sprayed on soldiers, and they were, at the time, they were seemingly quite proud and happy of it, to be exposed to it. It turns out to be a very toxic chemical, and mosquitoes were killed quite readily on contact with the material. We don't use this anymore. Once that got into the food chain, it was bioaccumulative, so we have outlawed that one. The easiest way really to get this under control, cheap mosquito netting. That's an inexpensive way to deal with it. There are malaria vaccines, there are chemicals that you can take to deal with the disease, but really what you want to do is avoid getting bitten to begin with. And when you're sleeping, it's the mosquito netting that makes all the difference. Now, malaria can, of course, kill quite readily because that fever, that excessive fever and those excessive chills is more than a lot of bodies can stand. You have to understand, young children, even adults can't take it. And imagine if you're in your twilight years, much older, and you get a serious malaria cycle, the fever chills, fever chills, fever chills, the body just gives up. So a malarial infection, you got a really good slide of it here from the bite where the sporozoite gets in. So on your diagram, you want to make a little note that this is your sporozoite phase. This is where it starts. And really up to this point, that's all about the sporozoite. And once it gets into your liver, we'll keep this simple because there's a lot to this. You get what's called the merozoite phase. If I can write. And the merozoite is when, well, let's put it this way. The sporozoite is in a more mature sort of radical form. This form, when released, when the cysts in the liver release the little critter that's broken out, the merozoites Well, they attack the red blood cells, and the red blood cells will pop as the parasite enters the red blood cell. It's found a new home to infect, and it's fulfilling its life cycle. It's heading back to its sporozoite-like existence. So it's going through a couple of phases just inside the body. I'd like to keep it about this simple because there is so much to it. But what needs to be delineated here, just so that we're clear, I'll go with blue this time, is you get a horrendous fever phase right up, well, if I had to draw a line, I'd say right through here. Let's push that back. There you go. You can't win for trying. So tremendous fever as the cells of the liver are infected. Where you would get the chills would be at this point, over here, as the white blood cells are popping. I always tell my students, imagine what it would be like, unfortunately, if you're bleeding to death, right? As the cells are being lost from the body. That's not really a far-fetched analogy. As your red blood cells are popping, they're dying, and you're losing a lot of that vital heat that you would need. So this alternation of fever and chills, there's a lot of parasite. So you got to realize the cells of the liver get reinfected, and the cells of the liver release these merozoites, which in turn attack your red blood cells. This can be brought under control with medications, for example, known as quinines. But the most important thing would be not to suffer an infection of this nature at all. So the subsequent slides here more or less just run through a bit of the information. Now remember, it's female mosquitoes, right? They're the bloodsuckers, and you're picking up those Plasmodium gamete cells. Essentially, they're going to get into your body at this point. And it provides a lot of background information here, but it's a little bit gross. The sexual phase did happen inside the mosquito, so yeah. So the sporozoites, they get through the skin, and it's the proboscis of the mosquito, which is the delivery device. It's one of the reasons I don't like mosquitoes. They're a vector for a lot of diseases, including West Nile virus. But the sporozoites would not have been able to get inside of our body if it wasn't for the help of the mosquito. And it is a fair question to say, well, what was infected in the mosquito? It's not just the gut lining, it's the salivary glands. So this is a good little slide because it shows the Plasmodium Sporozoites, and they're entering the liver, and they begin to, it's a great place, I mean, for food, if you think about it. There's nutrition flowing through the liver, your blood's flowing through it, oxygen and dissolved materials, and all sorts of things that these little critters can use to live. And at this point, you would be experiencing a tremendous fever, right? As your body begins to fight back. The liver is just ravaged by this disease. You can see why. How do you ever really get over it, right? If they form cysts and they take up home in the liver, what are the cues for an outbreak? Well, the outbreak could just be due to stress, get worn down, old age. But those infected liver cells, they burst, and the merozoites come flooding out, as we said before, and they go hunting down the red blood cells. So even though your body's fighting back, it's kind of a losing battle. So sorry, I'll just pause for a second. So at this point, we call them the merozoites. And once they find red blood cells to reproduce in, they bust out of those too. And this is the chill phase. So when it comes to malaria, people that suffer from malaria will go through these horrendous swings, fever chills, and you can understand as a medical practitioner just how hard that is to help them with. Right? At one point they're so cold that you're trying to keep them warm, and then at other times they're just bathed in their own sweat because of this disease, where the merozoites are affecting the liver and their body's fighting back. You get the idea. So the ecology of animal-like protists is pretty interesting. A lot of our animal-like protists will try to, well, live symbiotically within other organisms. And I think we just saw a case of that, a parasite, right? So put a little note here, for example, malaria, caused by Plasmodium Vivax, pretty nasty little critter. But it's not all bad. If you've got a lagoon, or if you even go down to the sewage treatment plant, there are protists, animal-like protists in there, and they're feeding on the dead organic matter, and that makes them, they're heterotrophic, but at that point, we'd say if they're feeding on dead organic matter, we say that they're saprophytes, or saprophytic. If you hunt down and kill something and eat it, I mean, that's heterotrophy, right? But being saprophytic, you're generally going after dead organic material. So the little cilia and the flagella of these organisms, as they're beating in, let's say, for example, a lagoon, right, where wastes are being processed, they're aerating that. They're not only consuming it, but they're helping to bring in more oxygen. That makes it so that more of these critters can live, and they process waste, and that's a huge ecological significant, sort of important position on our planet. So of course they themselves turn out to be part of the food chain. There's always a bigger fish, and protists are always eating protists, and larger things are even feeding on those larger things. Now, interestingly enough, these little guys, I'm going to zoom in here, are termites. So what are they doing? Well, termites are just an absolute nightmare. If you live down in Los Angeles, this is not what you want to see, or anywhere else in the world, if you're looking at the construction material for your home, because they can break down cellulose, and cellulose is a material that we find inside of wood. So let's make a little note there. So termites eat wood. I think we all knew that. But what if I told you it's not the termite that's actually eating the wood? You have to go a little bit further. You have to look a little bit closer. Turns out that there's a protist inside the gut, in the digestive system of the termite. So it's a symbiote. Okay, so let's make a little note on that. Symbiotic. Oh, you're gonna let me right here? I don't know. Symbiote. There we go. Trichonympha is producing the enzyme that lets our little friend here, the termite, consume this. So it's not the termite, it's the cellulase enzyme released by Trichonympha. From this guy. So it's the symbiote that's helping them to digest the wood. If you want to see this in action, you can use radiation, sterilize the gut of a termite, and the termite will starve to death. Right? The termite can't handle it, because what happens is it has no way to feed itself anymore. The symbiote is dead. Ooh, pop back in here. Alrighty. So without the symbiote, no life. And that takes us to the end of section 20.2. I'll post some neat stuff on my Twitter account related to malaria. I think there's a bit on the course website as well. But at this point, folks, take a break and say hasta luego. Have a good evening, day, morning, whichever it may be.